My name is Monica Rosenberg. I'm an assistant professor in the Department of Psychology at the University of Chicago. I'm really excited to be part of this workshop on naturalistic stimuli and individual differences, and to talk to you about my lab's work characterizing attention in the lab and the real world using functional brain connectivity. Now, during this talk, unfortunately, you can't interrupt me if you have questions, but if you do, please submit them through the online portal in the workshop website. You can do this until the end of August. Also, feel free to send me an email if you have more questions and want to discuss further. Have you ever had the experience of reading a paragraph in a book or a paper only to get to the end and have no idea what you just read? What about watching a movie or a TV show and realizing that you were distracted or mind-wandering and you lost track of the plot? We all experience these fluctuations in attentional state in our day-to-day lives. It's also the case that individuals differ widely in the ability that they have to maintain focus over time. Whereas some individuals have an easy time sustaining attention, they can maintain focus for long periods of time. Other people get distracted or mind wander more easily, more frequently. So how do we measure these individual differences in sustained attention and changes in attention over time? So this is a subset of tasks and questionnaires and clinical measures in the literature that I'm familiar with. What I want to propose in my talk today is that although these measures are really valuable ways to assess individual differences and intra-individual change in sustained attention, we can use another measure to complement these existing indices. And in particular, that's a measure from functional brain connectivity. So before I explain why I think functional brain connectivity is a useful measure of attention in lab-based and more naturalistic settings, I want to just get on the same page about how I'll be measuring brain connectivity throughout the talk. So in order to measure an individual's functional connectivity pattern, we collect functional MRI data while the individual is resting or performing a task or engaged in a naturalistic task. And we look at the blood oxygen level dependent signal in every node of the brain. That's what we call these individual regions of the brain. And we correlate signal time courses in spatially distinct parts of the brain. So for example, in order to measure the functional connection between this region of the brain or node in red and the node in green, we just correlate their signal time courses. And if we correlate the time courses of all possible pairs of nodes, that gives us this whole brain functional connectivity matrix, sometimes referred to as an individual's functional connectome. And so in this matrix, those cells in blue represent the correlation between activity time series in pairs of regions of the brain. We can project these functional connectivity matrices onto a brain figure like this. And I like to show this figure because it reminds me to emphasize that line reflecting a functional connection in blue isn't implying a direct structural connection between these two parts of the brain. Instead, it reflects statistical dependence between their activity time courses. So I started the talk by talking about attention. Why might these functional connectivity patterns be relevant to help us understand how people pay attention and why attention fluctuates over time? Well, work from a number of different researchers in the field has indicated that individuals have a unique pattern of functional brain connectivity that's relatively stable over time, and that we can use to identify people like a fingerprint. So this was work pioneered by Emily Finn, who you'll also be hearing from as part of this workshop. But unlike fingerprints, it turns out that we can actually use these functional connectivity patterns to gain insight into individuals' cognitive abilities and behavior. And so what I'll propose today is that we can use these functional connectivity measures to characterize how well people sustain attention and how their attention changes over time. And I think broadly, this kind of individualized prediction approach can really help us move fMRI from a science of group averages, which historically we've learned a lot by asking, what happens in the brain on average when people focus on certain information or try and maintain information in mind, to a science of individual differences. In other words, really asking whether we could use a single person's functional brain connectivity pattern to learn something about their unique patterns of behavior and cognitive performance. And so in the talk, I'll be going through a series of studies arguing that we can use functional connectivity-based models or connectome-based models to predict individual differences in sustained attention, to capture pharmacologically induced changes in attention over time, to capture dynamics of attention that occur in more sort of naturalistic ways rather than induced with pharmacological manipulation over time and in different contexts. And finally, I'll show some evidence that we can use these approaches to characterize not only sustained attention, but interactions between sustained attention and other cognitive processes like memory. And so to start off, I'll introduce you to a model that we built using brain connectivity to predict individual differences in sustained attention. So to build this model, we brought 25 healthy young adults into the lab, and we asked them to perform a challenging sustained attention task during MRI. I'm going to give you a little bit of a taste of this task now. You'll see the sort of experience that our participants had in the scanner. So in this task, participants saw images fading from one to the next. They were instructed to press a button every time they saw a city scene and to withhold a response every time they saw a rare target mountain, which only occurred about 10% of the time. And so, you saw a couple seconds worth of this task. In our experiment, participants performed the task for about 36 minutes in total with some breaks in between. And it was challenging. So even in our healthy young adult sample, we still saw a wide variability in performance. And so for each of the 25 participants in our sample, we could compute a measure of whole brain functional connectivity from data observed as they were performing that gradual onset continuous performance task, or GRAD-CBT. And we also had a measure of how they performed on the task. So here we operationalized sustained attention function as sensitivity, or D prime, on the task. This is highly related to percent accuracy and is computed by comparing an individual's hit rate to those city scenes relative to their false alarm rate to the mountain scenes. And so the goal of this project was to ask whether we could use functional connectivity patterns to predict individual differences in attention task performance. The way that we did this was with a technique known as Connectome-Based Predictive Modeling. This approach is described in a protocol published in 2017, led by Shilan Shen, and I encourage you to check it out if you're interested in applying it to your own data. I'll go through it briefly, and again, I'm happy to field questions through the website portal or over email. So in Connectome-Based Predictive Modeling, the first step is to retain a subset of data from our sample to use as a training set. And now we want to ask which possible functional connections in our whole brain functional connectivity pattern are related to sustained attention task performance, right? With the functional atlas we use, which has 268 nodes, we have over 35,000 total functional connections. So which of those are related to attention? To ask that question, we just perform mass univariate feature selection. So we correlate the strength of every functional connection with D prime scores across individuals in our training set. And so that gives us this matrix here. In this matrix, showing the relationship between task performance and functional connection strength, cells in orange indicate connections that are stronger in individuals with higher D prime scores, whereas cells in blue indicate connections that are stronger in individuals with lower D prime scores. If we threshold this matrix, we can retain the functional connections that are most strongly related to behavior in the positive direction. We call those the high attention network, and most strongly related to behavior in the negative direction. We call those the low attention network. And so now that we have these two sets of functional connections or edges that are related to behavior, we want to formalize the relationship between strength in these feature sets and task performance. And we do that by building a linear model that simply relates the degree to which each person expresses the high attention network and the low attention network on average to their performance score. And so in our training set, right, we see the expected result. Individuals who overall express the high attention network more strongly perform better on the task, whereas individuals who overall express the low attention network more strongly perform worse on the task. Now, this result is sort of guaranteed by our feature selection approach. And so how do we know we've identified networks that are related to sustained attention in general, and that we haven't just overfit a model in our specific training set of, here 24 individuals? To ask this question, we move on to the critical step of connectome-based predictive modeling of inputting data from an individual who we've never seen before. So now we look at the degree to which this left out 25th individual expressed the high attention network. We input that value into the linear model to get a predicted measure of performance. And we can do the same thing with their low attention network score. And that gives us a predicted measure of performance for this individual. And so if we use leave one subject out cross-validation, we can get a predicted measure of performance for each person in the data set. And now, to ask if our model has accurately captured sustained attention task performance, we can compare these predicted scores to actual task performance scores. And so that's what I'm showing you here. So on the x-axis is an individual's observed D prime values. So higher values reflect better performance on that continuous performance task. Now on the y-axis is predicted D prime values from functional connectivity data observed as the person was performing the task. As you can see, our model significantly predicted sustained attention task performance, right? Capturing over 70% of the variance in people's, how well people perform the task. And so this suggests, right, that we can look at an individual's functional connectivity pattern and predict with a fairly good, degree of accuracy how well they're performing on the task. I started this talk by proposing that we can use functional brain connectivity to help understand individual differences in sustained attention. But you might be thinking, right, if we have to give an individual a continuous performance task for about 36 minutes in the scanner in order to measure brain connectivity during the task, in order to predict task performance, like what's the added benefit of functional MRI here, right? Why didn't we just measure their task performance with their button presses? To ask this question, we performed a subsequent analysis where now we asked, do we actually need to have individuals perform a sustained attention task during scanning in order to characterize their sustained attention function? Do we actually learn something about their sustained attention abilities from looking at data observed while they're not performing any attention task at all? To ask this question, we took our models defined on 24 individuals' task data and applied them completely unchanged to left out to resting state data from the held out individual. And what we can see is we still get accurate predictions of performance, suggesting that we don't actually need to tax individuals' sustained attention function in order to predict their overall sustained attention abilities. Instead, the functional networks that are related to sustained attention seem to be online and informative, even when people are just resting quietly in the scanner. And now, interestingly, we can see that our predictions from resting state data are numerically worse than our predictions from task data. And we think this may be because when we give people a sustained attention task, we're actually taxing the neural circuits related to sustained attention and potentially magnifying these differences in functional connectivity that are related to individual differences in sustained attention. So, these data are good evidence that we can use functional connectivity to predict attention task performance. But this is a workshop about naturalistic stimuli and the importance of considering cognitive and attentional processes in the real world. So the question came up, right? Have we built a model that captures sustained attention function in general? Or have we built a model that captures performance on this, somewhat idiosyncratic lab-based task of attention? To ask that question, we applied these connectome-based models completely unchanged to a totally independent data set that was almost as different as possible from the data set that I just showed you. Now, this was a functional MRI data set of children and adolescents with and without symptoms of attention deficit hyperactivity disorder. So attention deficit hyperactivity disorder is characterized by inattentiveness, hyperactivity, and impulsivity. In other words, deficits in sustained attention. The worldwide prevalence of ADHD is estimated at 5 to 7%. And the costs associated with ADHD are huge, right? They exceeded $36 billion annually more than 10 years ago in the US alone. So to ask whether our model generalizes to a totally independent developmental fMRI dataset to capture this real-world measure of sustained attention function, we looked at a publicly available dataset, the ADHD 200 sample. This sample, as I mentioned, included functional MRI data from 113 children and adolescents between the ages of 8 and 16. For each of these participants, we had a measure of their resting state functional connectivity pattern. So the participants in this sample didn't perform any sustained attention tasks. We only had resting state fMRI data. For each of these individuals, we also had a clinician-rated measure of ADHD symptom scores. So we had this continuous rating for individuals who did and who did not have clinical diagnoses of ADHD. And the goal of the analysis was to take our model defined on our totally independent adult data set, apply it to each of these resting state connectivity patterns to predict how, if we hypothetically gave the continuous performance task to each child or adolescent in this data set, how well they would perform. So what I'm showing you here on the x-axis is observed ADHD symptoms. So higher scores represent more frequent or more severe ADHD symptoms. And on the y-axis is predicted sustained attention task performance. So as you can see, we see the expected negative relationship, such that when a child has frequent or severe ADHD symptoms, the model actually predicts that that child would perform worse on the attention task. Now, this is evidence that the model is generalizing across data sets, scan sites and parameters, participant populations, and measures of sustained attention, right? From this lab-based measure of sustained attention to this real-world clinical measure of attention function. But is our model capturing sustained attention, or is it just capturing sort of cognitive abilities in general? In other words, it's generalized, but has it generalized too far? Is it really specific to sustained attention function? To sort of investigate this further, we related the predictions of our model to individuals' ADHD scores when controlling for a measure of fluid intelligence, and we still see a significant relationship. However, when we compare model predictions to fluid intelligence scores when controlling for ADHD symptoms, we don't see a significant relationship. In other words, demonstrating that the model is generalizing to capture different measures of sustained attention, but isn't capturing sort of cognitive abilities in general. Instead, is relatively specific to sustained attention function. This is evidence that the model is generalizing across, right, participant populations, as I mentioned, from an adult population to a developmental population. And I want to just show a little bit of new evidence from my lab, demonstrating that this result holds in an even larger and more heterogeneous developmental sample. And so this is work led by a postdoc in my lab, Dr. Omid Kardan. And Omid looked at functional MRI data from the large publicly available adolescent brain cognitive development study sample. Now, this is a functional MRI data set with more than 10,000 individuals collected at 21 sites across the United States. In this sample, participants perform an NBAC task during fMRI, and some blocks of the NBAC task are a zero-back task. In this task, participants are shown a target image, so either a face or a scene, and then they're shown a series of other images, and they just need to indicate whether every image they see matches the target image or doesn't match the target. And so this zero-back task has a low working memory load, right? Participants need to remember the target image and mentally compare it to every new image that they see. But it's also an attentionally taxing task, right? They need to maintain focus on the task in order to maintain good performance. We also see a lot of similarities between this zero-back target detection task and a traditional continuous performance task used to assess sustained attention, like the GRAD CBT with cities and mountains that I introduced earlier. And so our question was, in this large heterogeneous developmental sample in which children perform the zero-back task, sort of akin to a sustained attention task, does the model capture individual differences in performance? Again, the same high and low attention network model that we defined in our initial adult data set and generalized to predict ADHD symptoms in the ADHD 200 sample. So to ask this question, Omid looked at functional MRI data during zero-back task performance in about 1,500 participants who underwent extensive visual quality control and motion exclusion thresholds. And what he found was that indeed the model did generalize to capture performance, such that individuals who performed better on the zero-back task were predicted to have better sustained attention function. So there's just more evidence that the model sort of externally validated across independent data sets to capture individual differences in different measures of sustained attention. So I've talked a lot about this predictive model, the high and the low attention networks that generalize across data sets to capture individual differences in sustained attention. What are these networks? You might be curious what they actually look like. I'll sort of go through briefly their anatomy here. So in orange, you can see the high attention network, and in blue, you can see the low attention network. Spheres represent nodes of the network, and the spheres are sized according to the number of functional connections they have in both networks, and they're colored according to the network in which they have more connections. And so interestingly, you can see that there are some large spheres that appear gray. And that's because these particular nodes have approximately equal number of functional connections in the high attention network and the low attention network. And I think this just emphasizes the importance, especially when predicting individual differences in attention, of considering interactions between multiple regions rather than looking at activity in single regions in isolation. If you're interested in diving further into the anatomy of these networks, or you want to apply them to data that you've collected, feel free to check them out. They're available on GitHub, as is code that can be used for applying them to new data sets. Another way that we can visualize the network that I think gives sort of a big picture overview of their anatomy is with these circle plots. So here in the circles, the left hemisphere of the circle reflects the left hemisphere of the brain, and the right half of the circle reflects the right hemisphere of the brain. And the nodes of the network are organized according to macro scale brain regions. So for example, the prefrontal nodes at the top, motor nodes next to them, and so on. And you can see some sort of gross anatomical trends. For example, frequent intracerebellar connections reflected in the low attention network, potentially related to this finding of sort of common cerebellar changes in individuals with ADHD. And so I hope this series of studies has convinced you that looking at individuals' differences in functional brain connectivity can give us insight into individual differences in sustained attention function. But I want to move on to a really important question, which might actually be relevant at this part of the talk, which is sustained attention isn't just a single solitary trait. It's not always the case, right, that individuals with successful sustained attention function are always attentive, whereas individuals who struggle more to maintain focus are always inattentive. Rather, our attentional state fluctuates over time. It might be better on some days and worse on other days. It might be better during some moments of a virtual lecture like at the beginning, and it might wane as we go into the talk. And so, is it the case that all individuals just have this static functional connectivity pattern that reflects attention, and there's nothing we can do about it, right? It's just, that's how it is and it doesn't change. Or is it the case that these functional networks actually change with changes in attentional state within individuals? As an initial way to ask whether this was the case, my collaborators and I looked at data from individuals who had attentional states modulated by pharmacological interventions. So the first way that we sort of looked into this further is we looked at data that had been collected by collaborators. These data were functional MRI data collected while individuals were resting and performing a stop signal task. And some of the individuals in the data set had been given a single dose of methylphenidate or Ritalin before an fMRI scan. So methylphenidate is a common and effective ADHD drug. And our question was, do individuals that had been given a single dose of methylphenidate before fMRI show functional connectivity signatures of stronger sustained attention? When we compared high attention network strength between the individuals given methylphenidate and a different group of unmedicated individuals, we saw that indeed individuals who had been given methylphenidate before the scan showed stronger high attention networks. Again, these are the same networks defined in our totally independent data set. Interestingly, when we looked at strength in the low attention network, again, this is a network that's stronger in people who performed worse on that sustained attention task, we see the opposite pattern of results, such that individuals given methylphenidate show weaker low attention networks. And so it's not just the case that methylphenidate is having an overall effect on whole brain functional connectivity. Instead, it appears to be selectively modulating connection strength in networks that are related to sustained attention. These data I'm showing you are from rest, but we saw the same pattern of results when we looked at the stop signal task data. Now, I think this is an interesting result, but one sort of limitation of this study is that this is a between subjects manipulation, right? We don't have functional connectivity data from the same people who had been given methylphenidate and hadn't. And so in order to ask whether these networks are malleable within single individuals, in other words, whether they change with changes in attentional state in the same person, we looked at data from 2 other data sets collected by collaborators. And now in these data sets, individuals weren't given a pharmacological intervention that improved attentional state, but instead they were given a pharmacological intervention that had the opposite effect. And so these were anesthetic agents. And so in these data sets, individuals were given either a dose of propofol or sevoflurane. And anesthetic agents have many other effects in addition to modulating attentional state, right? They affect other cognitive states and states of consciousness, but one of the processes that they do affect is attention. And so we asked, do individuals scanned while awake and just resting in the scanner show functional connectivity signatures of stronger sustained attention than they do while they're under deep sedation with propofol? And so here, every line I'm showing you is a single individual, connecting points indicating their high attention network strength while they're awake on the left or under deep sedation with propofol on the right. These results are from the high attention network. We see the opposite pattern in the low attention network, demonstrating that in nearly every individual in this sample, it is the case that they show connectivity signatures of stronger attention when they're awake than when they're under deep sedation. We see a similar pattern of results when we compare strength in these networks while people are awake and resting and while they're under light anesthesia with sevoflurane and recovering from anesthesia. And so again, these data provide evidence that the networks are malleable within individuals, and they're not just a static trait and you're born with a certain network strength and you can't do anything about it. Recent work from Taylor Chamberlain, who's a lab manager in my lab, actually pre-registered a hypothesis that we would replicate these results in a different data set that's publicly available on OpenNeuro. And so really interestingly, this data set has functional MRI data from people who are awake, recovering from anesthesia and under deep sedation with propofol. And they have these data collected while people were resting during MRI scanning. And they also have these same conditions collected while people were engaged in a naturalistic task, while they were just listening to a narrative, which was the dialogue from the movie Taken. So it was like a short clip from the movie. And what Taylor found was in this independent data set collected by sort of a different research group, we replicated the effect such that high attention network strength decreased as anesthetic state increased, and low attention network strength increased. In other words, right, people showed stronger functional connectivity signatures of stronger attention when they were awake. So this set of studies, I hope, is sort of providing some evidence that these networks are malleable and they change with changes in attentional state in single individuals. But I motivated this part of the talk by appealing to our phenomenological experience that we all experience fluctuations in attention. We know that we're not equally focused at all moments of time. But it's probably not the case that the most common way that we experience attentional state fluctuations is with pharmacological interventions. Rather, attention sort of naturally and dynamically changes over time in many different parts of our lives. So, as I talked about earlier, reading a book or a paper or watching a movie or, doing an attention task in the lab. And so isn't the case that the same functional networks that capture individual differences in sustained attention also fluctuate with these sort of moment-to-moment, day-to-day, week-to-week changes in our attentional state? We took advantage of a really unique data set that's available on OpenNeuro in order to sort of start asking this question. In this data set, the same individual was scanned 30 times over the course of about 11 months. And during every scanning session, this individual performed one run of the Grad-CPT, that city mountain task that I introduced earlier. And so our question was, do the same functional networks that predict individual differences in sustained attention actually vary with session-to-session task performance in the same person? So what I'm showing you here on the x-axis is observed D prime or sensitivity during each of those 30 task sessions. And on the y-axis is a model prediction for each session separately. And so again, this was the model defined previously, the same high and low attention networks that I've been talking about. What we find is that indeed the model is sensitive to this person's session-to-session change in task performance. So it's not just the case that it's capturing sort of a person's overall or mean average sustained attention function, but it does seem to be sensitive to these session-specific changes in attention task performance. What about higher frequency, sustained attention fluctuations, the kind that we might experience within the course of a single lecture, not the kind that happen, over the course of several months? We took advantage of a feature of the data set that I introduced in the first part of my talk in order to ask whether the model is sensitive to sort of higher frequency changes in attention. In particular, in that 25-person data set, participants in the scanner performed 3 minutes of the continuous performance task. They took a 30-second break, performed the task for 3 more minutes, and so on, until they had performed the task for 12 blocks in total. So for each of the participants in this data set, we have 12 blocks of sustained attention task performance, meaning that we can calculate 12 distinct functional connectivity matrices just by looking at data during those 3-minute task blocks separately. We can also measure task performance in each task block separately. And sort of now the question is, can we take those functional connectivity models defined in 24 participants' task data and apply them to functional connectivity from each of the left out person's task blocks separately to get a predicted measure of performance for each of those 12 blocks? And now we know that in these data, functional connectivity captures a person's overall task performance, but if it's also sensitive to which of their task blocks were better and which were worse, right, which they struggled to perform well in, we should see a high positive correlation between predicted block performance and actual block performance. So that's what I'm showing you here. In this graph, every bar is an individual participant, and the y-axis, or the height of the bar, corresponds to that within subject correlation. So a correlation of 1 would mean that the model was sensitive to, which were the person's best task blocks and which were their worst. And a correlation of 0 would, suggest that, well, we know that the model is capturing the overall task performance, but it's not really sensitive to these, fluctuations in task performance from one 3-minute task block to the next. So what did we find? In these data, we indeed found that on average, the model captured block-to-block changes in attention task performance So this effect was significant at the group level. And model predictions also significantly predicted block-to-block performance changes in individuals whose bars are shaded in gray. So this is some evidence that these high and low attention networks are changing in their strength with changes in session-to-session sustained attention task performance and minute-to-minute sustained attention task performance. It's not the case, as I've emphasized, during this talk, that our attention only fluctuates in the lab when we can measure it with really high resolution behavioral measures, and we can use specific tools from psychology in order to characterize those fluctuations. Instead, these also happen in the real world. And do these networks actually capture fluctuations in attentional state that happen in other more real world contexts? Like, do they track how our attention changes during a movie or as we listen to a story? I want to talk about some recent work in my lab using this approach to predict fluctuations in narrative engagement. In other words, how we attend to movies and stories. This work is led by a PhD student working in my lab, Hayoung Song, and is currently in press. So if you're interested in the work, I encourage you to check out the paper, which should be coming out this week or next week very soon. So in this project, Hayoung was really interested in asking whether changes in functional connectivity strength capture changes in attention fluctuation in naturalistic narrative contexts. In order to do that, she used two fMRI datasets that are openly available in the field. So the first is a dataset collected by our collaborator, Emily Finn. This was a sample of 22 individuals in the study. Each individual listened to an audio narrated story during fMRI. And the story was purposely written to be suspenseful and to have sort of mysterious twists and turns. In the other data set provided by Janice Chen and colleagues, individuals watched the first episode of the BBC TV show Sherlock during fMRI. And now the question was, can we use changes in functional connectivity patterns to capture changes in attention in these two data sets? But the problem was, these data were collected years ago, and we don't have a measure of how people that participated in these studies were actually attending to the narratives during fMRI. So how could we measure how attention capturing these different narratives were? In order to do this, Hayoung came up with a pretty clever approach to characterize how subjectively engaging these two narratives were. She brought new behavioral participants into the lab at UChicago, and she just asked them to either listen to the audio narrated story or to watch the Sherlock TV episode and to rate how engaging they found the narratives. And so in particular, she told participants, during this task, you'll listen to a story. You will continuously rate how engaging you find the story. You can press the left and the right keys to adjust the scale. So, as they were listening or watching, participants could continuously adjust a scale bar to indicate how engaged they were in the narratives. We gave them a little bit of context about what it meant to be sort of engaged in those narratives. And in particular, we told them that they find the story engaging when they're curious and excited to know what's coming up next, when they're immersed in the story and their attention is focused on the story, but they don't find the story engaging when they're bored or other things pop into their mind or their attention wanders away from the story. So this is could be a pretty difficult task. We know that sometimes individuals are aware of their own mind wandering, but other times they're completely unaware of when they're mind wandering. So is it the case that people can give us accurate reports about how engaged they are in a narrative? Or is it the case that this sort of dual task, watching the narrative and rating your engagement, is difficult, and so we don't really see consistency across individuals and their engagement ratings? What I'm showing you here is the data from these behavioral participants. So every row is data from a single individual, and the cells, the color of the cells indicate how engaged that person said they were at a particular moment of the story. And as you can see, in this audio narrated story, changes in subjective engagement were highly synchronized across individuals, There appear to be distinct moments of the story when everybody reported finding the narrative very engaging, and other moments when they reported that it was less subjectively engaging. We see similar consistency in the Sherlock TV show. If we plot the group average engagement ratings with 95% confidence intervals, again, we not only see that engagement ratings were relatively synchronized across individuals, but they were also highly variable over time, right? As I pointed out in the other figures, there's moments of time that are more engaging and moments of time that are just less engaging, that everybody finds less attention grabbing. We can qualitatively assess these engagement ratings by comparing, asking what's happening in the narratives during peak moments of engagement. So what about this peak in the audio narrated story? We can see, right, that everybody was more engaged on average when there was a mysterious disappearance of a character or a twist ending in the story. And in the Sherlock TV show, I hope I'm not spoiling this, but we see, right, a relatively consistent moment of high engagement when Sherlock discovers a corpse, which is a clue in the mystery that he's trying to solve. So now that we have these measures of group average engagement, we can sort of use them as a quote unquote ground truth measure of how attentionally engaging the narratives are over time. And we can ask, do changes in attention network connectivity, so the same sustained attention networks that I've presented earlier, predict changes in group level engagement over time? So to ask this question, Hyung used an approach that she first described in a preprint from last year that's available if you're interested in learning more, where she performed dynamic predictive modeling of these group level engagement measures. In particular, she's trained support vector regression models to predict engagement at every moment of time in a subset of participants, and applied those models to data from held out individuals to predict changes in engagement. And so what she found, I'm first going to show you data from the within dataset prediction. So in other words, training support vector regression models using patterns of connectivity in the sustained attention networks that I've already presented to predict changes in engagement over time. Interestingly, the results revealed that in the audio narrated story, we call this the paranoia dataset, patterns of connections in the sustained attention network didn't capture dynamic changes in engagement over time. However, in the Sherlock dataset, sort of multivariate patterns of connections in the sustained attention network did significantly predict changes in engagement over time. Now, there's a lot of reasons we could see differences in the pattern of results between these two data sets. They have different numbers of participants, they were collected at different scan sites with different scan parameters. One hypothesis is that the sustained attention networks are better capturing engagement in the Sherlock dataset because they were defined to predict performance on a visual sustained attention task, and Sherlock has visual information, whereas the audio narrated paranoia story is only auditory, right? It may be the case that there's distinct networks that capture visual and auditory sustained attention. This is a completely speculative hypothesis at this point, but we're conducting further studies now to follow up on this question. So it may be the case that networks that predict sustained attention and narrative engagement in naturalistic contexts are partially, but not completely overlapping, right? It could be that attention during narratives relies on a overlapping, but somewhat distinct set of functional brain networks. So to ask if this was the case, Hyung performed dynamic predictive modeling again to define new networks that predicted changing engagement in the paranoia and the Sherlock datasets. And she found that in both cases, multivariate patterns of connectivity did indeed significantly capture changing engagement over time. Furthermore, when she used the support vector regression trained to predict engagement on the paranoia dataset and applied it to predict engagement in the Sherlock dataset and vice versa, she saw significant prediction of engagement in both cases. In other words, these models were generalizing across completely separate datasets and different narratives to capture changes in engagement, right? They really seem to be capturing changes in this group level engagement measure, and not something story specific or specific to the low level features of one of the narrative stimuli. So I've gone over evidence, right, that the same functional networks capture dynamics of attention over time and across contexts, right, more lab-based and naturalistic narrative contexts. And I want to show a little bit of evidence here in the last part of my talk that we can use this approach not only to capture inter and intra-individual differences in sustained attention, but to actually understand how sustained attention interacts with other cognitive processes, and in particular, our focus on memory today. So, what are the consequences of paying attention to a naturalistic stimulus for what we remember? To start asking this question, I collaborated with researchers at the NIH. This study was led by Dave Jangra, who's now a faculty member at the University of Vermont. And this was a really cool fMRI study in which participants were scanned as they were reading transcripts of complicated Greek history lectures. So these were college level lectures about Greek history, and participants were screened beforehand to make sure they weren't experts in Greek history. So they read these transcripts during fMRI, and then after they got out of the scanner, they were given a test to assess what they remembered and what they recalled from what they read. And what the researchers found is that the degree to which these individuals had expressed the high attention network relative to the low attention network was related to their performance on that recall test. In other words, individuals who showed stronger functional connectivity signatures of sustained attention during reading better recalled what they read after they got out of the scanner. Now, this is some evidence, right, that our attentional state during reading has consequences for what we later remember, but it could also be explained, from an individual differences perspective, that people who tend to pay attention more during scanning tend to also remember more, just in general, whenever given a recall test. So is it the case that, within people, moments of time when we're more attentive are also, related to better memory for the information we encounter when we're more attentive? So returning to the study that I talked about, using dynamic changes in connectivity to predict dynamic changes in engagement, we started to ask this question, right? So again, looking at the two narrative data sets that I introduced before, the paranoia audio narrated story data set and the Sherlock TV show data set, we asked whether multivariate patterns of connectivity in the same networks that predicted participants' engagement to the narratives also predicted their recall fidelity. So, after the participants listened to the story or watched the TV show during scanning, they performed a free recall task when they got out of the scanner to just, recall everything they could remember about those narratives. What we found was that in both cases, multivariate patterns of connectivity within the engagement network predicted participants' later recall dynamics. In other words, the same networks that predicted group level engagement also predicted individual recall time courses, suggesting that the degree to which we express networks that are related to attention and narrative engagement has consequences for what we later recall. And I encourage you to check out this paper if you're interested in learning more. So today, I've argued that we can use functional connectivity-based models to predict individual differences in sustained attention and to capture changes in attention over time, changes that are due to pharmacological manipulation, but also changes that occur more naturally as we perform attention tasks or listen to stories or watch movies. I've also shown some initial evidence that signatures of sustained attention have consequences beyond in-the-moment task performance, specifically impacting what we later remember. And so just to zoom out, I've argued that functional brain connectivity can be used as a brain-based biomarker of sustained attention across different participant populations and data sets and contexts. And furthermore, that the same brain networks predict differences in attention both between people, but also within the same individuals. And finally, network signatures of sustained attention predict subsequent memory. And so, I hope that we can begin to apply these individualized prediction approaches to, understand what's happening at the level of single individuals, right? How well does a single person pay attention? How does their attention change across different contexts, in addition to powerful group-based approaches that have historically been common in fMRI research? And so with that, I'd like to thank my funding sources and my awesome lab and my collaborators. And thank you so much for joining me in this virtual workshop. I hope to hear from you in the Q&A portal and also over email. Thank you again for your attention.